the biggest cell of our body, which is the egg, and then it needs to merge with one of the smallest cells in our body, which are the sperm that you can see here in the background. And today I'll talk actually only on the sperm. And I like this to start with this image, not just because it's one of the first images we ever got on the microscope, but because it illustrates some of the challenges that we had to address over the years. And to go more native, I went to specialize in cryo-electron tomography. And then I summarize kind of the four problems or challenges that we have to deal with when doing cryo-electron tomography. And the first one is the sampling. So as you can see, this is not one image, but it's actually made of each of these squares is a high magnification image of part of the cell. So it's a general problem that you have with high-resolution imaging, that if you want high resolution, then you need to go for very high magnification. The other problem is the sample thickness that was already mentioned, that we are limited of the thickness of the samples we can look at, and you can see it here in the head. Obviously, the head of the sperm is not, it doesn't have a hole, but we didn't bother to image it because the electrons cannot go through this part of the cell. The third one maybe is a little bit less obvious just from this image, but what I actually like about tomography is that we see everything, we do not label anything, but that also comes with the challenge to being able to understand what we are looking, what do we see. And the last point is resolution. Was again already mentioned today that tomography is not necessarily the resolution revolution when people talk about it. It is referred to single particle analysis. It's something we have to deal with. It was already shown earlier today that it is getting better, but what I also want to show you that it's not always about the resolution. Okay. So with this kind of challenges in mind, this is where we started our journey and looking at sperm. So this is kind of an overview of the workflow that we are using to address and being able to study mammalian sperm. So what you see here is how the sample can look like. This is an example from boar sperm. We just put it on an EM grid. This is an image that was taken in a light microscope. So for light microscope, sperm are actually relatively small cells. And then we just plunge freezing, so it's a simple freezing. If we do want to localize specific area of the cell, then we will first do some labeling, and then we can do cryofluorescent microscopy. And then depending on which part of the cell we want to look at, we will either go directly to the transmission electron microscope if we look at the thinner part of the cell, or we will go through the focus ion beam milling to thin the sample. And then from the beginning, we wanted to really take advantage of the tomography on cellular sample, which means that once you have the sample in the microscope, you can look at every part of the cell. And so we took kind of a really holistic approach where we looked at all parts of the cell. So I can essentially take you through a walk along the cell, and we can start from the really end part, and it's called the end piece. This is a slice from a tomogram, and in this part of the cell you will find only singlet microtubules. And for the sharp eye in the background that know microtubule, you can see that these microtubules actually are not empty. We'll come back to that. And this is the plasma membrane that envelops the whole cell. But we can go, we can continue with our walk, and if we get closer, so this is the part of the tail that is called the principal piece, but this is kind of more distals in the principal piece. And now the structure becomes a bit more complex. What you see here in the center is the main motor apparatus of the cell, which is called the axoneme, that by itself is made of two singlet microtubules that are called the centroper that run in the center. You have the doublet microtubules from two sides, because again this is a slide from a tomogram, you can see. Then there are a structure that you call a fibrous sheath from two sides, and then again the plasma membrane. What you see here is a cross-section, but this is of course a tomogram, so I can actually play it so you can see the whole volume. But we can continue on, getting closer now to the head, so this is where it's becoming even more complex, and also it means thicker, so the sample is getting thicker, because now the axoneme that is in the center is also surrounded by the mitochondria, so the mitochondria is forming actually helix around the whole axoneme, so it's, I think, a really beautiful structure on the organelle level, if you want. So, and again, it's a tomogram, so I can play it too, so you can really see it's individual. It's not one mitochondria; it's individual mitochondria that wraps all around. And we can get even closer to the head. So this is where the head will be. And in this part of the cell, this is where you will find the centrioles and these structures that are called segmented columns. So as you can see, by now I have actually overview of a whole cell from beginning to end, or in my case I call it from head to tail. But obviously I don't have the time to tell you on all the parts of the cell, so I just chose two interesting, I think, stories to tell. The first is the centrioles, and the second part is going to be on the axoneme, that I mentioned is the motor apparatus of the cell. So let's start with the centrioles. So centrioles, as you might remember from your cell biology classes, it's a very important organelle to have in every cell. And its number is very well regulated, so every cell should have two centrioles. And these are the ones that also form the base of the flagellum. They have a very clear structure, so it forms this ninefold symmetric barrel that is made of triplet microtubules. So you have 3 microtubules, and you see 9 cones. So this is how centrioles should look like. But again, when it comes to fertilization, it has a very interesting story, because oocytes, as big as they are, they actually lose their centriole during their maturation. So eggs do not have centrioles. Then you would expect that the centrioles should come from the sperm. But when you look at classical electron microscopy, then started a problem, because mouse, when you look, do not have anything that resemble any centriole. When you look at human sperm, and this is already one example for you, differences that you have between species, because sperm are one of the most diverse cell types you can find. So there is something that looks like a barrel, which is close to the head, and this is why it's called a proximal centriole. But then this is only one, which is again raising a problem for fertilization, because the first cell division still needs to have centrioles. So our collaborator, Tomer Avidorais in the US, actually did some nice super resolution fluorescent microscopy, and found that you can see 2 parts where you will see, you can localize and find centriole proteins. But you can see that the other one, it looks already quite strange. And they did also correlated like in electron microscopy, and they could see another signal for more proteins. So then we looked closer in our tomograms, and this is again the part of the cell where you should find the centrioles. And what we found that indeed we do find in this region, and this is the segmentation that you can see here on the right, one barrel that has triplet microtubules, but then not all the microtubules are the same length. And then the other part, so we did find a centriole, but it's just a bit of a strange centriole, because now we see doublet microtubules and not triplet, how it should be. And we analyzed it a bit further, and we actually looked at it in different cells. So it is representative, so in the different cells, we saw it's always the case that you have, for the proximal centriole, triplets that are not the same length, and we did some subtomogram averaging that you can see these triplets here. We also found that these have microtubule inner proteins, unlike other centrioles. And then these distal centrioles, as I mentioned already, it's made of doublets, but also it doesn't form really a barrel, because the microtubules are really opening up. And that is a feature that we did see that is conserved between the different species that we looked at. And then so you might ask, so how do you know that it's actually a centriole and not the axoneme? So because we have the data from all parts of the cell, we actually captured also the transition from the centriole into the axoneme. And there are very specific characteristics that you can see here. So first of all, the overall geometry is different. So the axoneme is very symmetric, and it's really look like a barrel, but the centriole part, the microtubules are really opening up. But I think the most obvious one, if you want, is that you can see that the accessory proteins that known to be in the axoneme, you will not find them in the part which is the centriole. Okay? So I think this is a nice example, I think, of the power of tomography, and to show you that, yeah, sometimes you don't need to have high resolution to have important observation. And so the second part, I want to go back to the axoneme, which is the motor part. So we will look at more detail at the axoneme. And I like this drawing very much because it is based on classical electron microscopy that was done many years ago on sperm. And this already illustrates the complexity of mammalian sperm, which is very different from sperms from kind of other species. The model that was used before, it's sea urchin sperm, but there it's very simple. You just have the axoneme running all along the tail. But in this case, you can see in this drawing some of the data that I also showed you, this mitochondria that is around this axoneme, and this out of dense fibers and the fibrous sheets that are around. And then in this case, so we also got the subtomogram averaging of this structure. So you can see here, this is the central pair, which is in the middle, and an average of the doublet, which is kind of in the surrounding. But kind of a problem that we run into this, and it's very often a question that I would get, is that it's very nice, very colorful pictures, but which protein is it? And what you can see that the microtubules actually, they are full of proteins. And this is a question we were not able to answer with the tomography. And this is why we moved on and took, I hope I didn't do anything wrong. Okay. Maybe the projector is also getting tired, so I'll speak even faster. Yes. But I just want to highlight that some of the techniques that you might use in other culture cells that are actually not possible working with sperm. First of all, sperm are transcriptionally and translationally inactive, so you cannot do all of this genetic manipulation that you will do in a cell culture. Talking about cell culture, we cannot actually culture sperm, so they are good for a certain limit of time. We cannot keep them going in culture for too many days. And I already mentioned that there are very big differences between different species, and even if we go for transgenic animals, then the only one available are really mouse, maybe rat, yeah? And to add to the complexity, many of the proteins, they are really specific ones. So it's again, even if you do it in a cell culture, it's not necessarily going to be relevant. So yeah, many problems. So we had to be smart about going and trying to identify some of these proteins. And the inspiration actually came from, again, going back to this first image, and now I kind of show you the big version of this end piece here. And what you can see in this image is something we try to avoid and kind of try to fight against for 3 years, is that the cell actually lost the plasma membrane around it, so it's not here anymore. So for 3 years, when we did all this tomography, we tried to fight hard and find the condition, how to plunge that we don't lose the plasma membrane, and I showed you that we are able to do it. But the cell point we realized actually we can make use of this because what you can see here that now that the plasma membrane was lost, the microtubules actually kind of spread. And then when they spread and they are not overlapping, you no longer need the tomography, because then you can do single particle analysis. And that's exactly what Miguel in the group did. So we intentionally then now a lot longer to remove the membrane. And then we had very beautiful singlet microtubules that are ready for single particle analysis. By no means this is normal single particle analysis. We cannot use it here to collect the data. This is all done on serial EM. Because the sample is not everywhere, it's still the case that the whole cell is here, so we will not have our sample everywhere, okay? So it's much more time-consuming in terms of data collection, but very rewarding, because now we got to a resolution that we can essentially resolve the structure to better than 3.5 angstrom resolution. And we identified this protein. This is the called SPACA9, and because we had this very high resolution, we actually found that there is even another microtubule protein that we did not realize, which is called SAXO1, and this is a very thin one. It is not very structured actually, and it goes and it's running along each of these protofilaments. So this is the case for the singlet microtubules, and now the question is, that was perhaps the easiest part. The next question is, are we good enough, and how can we do it and approach really the axoneme? Because I show you this is not as simple. There are many more structures actually holding this all together. I showed you already this movie. So for that, Miguel had to be even more smarter, but essentially he went through the literature and found a very old publications that studied the function really of the axoneme. And in this protocol, you essentially disintegrate the whole axoneme by, first of all, of course, removing the membrane. We do it with the detergent, then we do some freeze thaw, and then we give ATP, because the motor proteins are the dynames and they need ATP for movement, and essentially the doublets slide along each other, and essentially we are disintegrating the whole axoneme. Yeah? So then we end up with something like that I think looks like more octopus rather than a sperm. But you can see here in a higher magnification, you can see that we have the doublet microtubule, and then the outer dense fibers that bound to it, they are not lost, they are still there in the sample. So again, this is a sample that is ready for single particle analysis data collection, and this is what we did, and with quite a bit of processing and classification for different parts of this doublet. You can see the local resolution that we got to the different parts of the doublets. So it means that now what we have is a map that is better than a 3.5 angstrom resolution all around. and this is kind of the improvement when you kind of zoom in, and then now every protein here is colored in different color. So we essentially got to the point that now this stopped working. Okay, yeah, we got to the point that we essentially identify most of these proteins. In this case, it's more than 65 proteins that fill the whole lumen of the A tubule, or most of it, and there are some in the B tubule. This is a cross-section, and this is kind of longitudinal section. You can see that many of the proteins that were identified were not kind of recognized before. They had even a strange name, and we just renamed them. They had like off name, no one knew what they are doing. But now we actually know exactly where they are in the structure. What you see is on the right is it's still the case in the field that the reviewers really wants you to prove by seeing the map for each of these proteins that you identify. And I can tell you for 65 proteins, it's a very big packet now that you can get it in our supplement. For each of these proteins, we had to create this. Yeah, I'm talking about big, yes, so this structure has close to 1.5 million atoms, which has 182 protein residues. So the best is actually to look, I think, in a movie, because then you can get the feeling of the whole structure. So what we're going to do next, and the white are the microtubules that are the scaffold, if you want. And this is the cartoon presentation of this model. And then we can remove the microtubules, so you can see how this structure is all over interconnected between the different proteins. Again, I do not have the time, and don't worry, I'm not going to talk about all this protein, but I just showed one example which I think is super interesting, specifically for structural biology. I think came up to be very interesting, which is the protein that fills the microtubule, the A microtubule. But what I want to show is that in parallel to our structure, there was a group in the US that did the structure from sea urchin sperm that I already mentioned, that is much simpler, has only the axoneme. But what you can see that also on the molecular level, it's much simpler, so it does not have many of these extra microtubule inner proteins that we find. So we, by comparing these two structures and other cilia, we actually came up with some core microtubular inner proteins. But the interesting bit, as you can see, is that what is happening in this A tubule, and what turns out to be is that mammalian sperm extended one of these proteins that is called tectin, and it has the tectin 5 filling most of this A tubule, and you do not have it in the other cilia or in the sea urchin sperm. And I think it's an interesting case for structural biology, because again, I'll show it in this movie, that we will focus now only on this tectin. So I said that these are all the tectin that fills the foam. So you see they form this filament inside the A tubule. So the 1 to 4 are the ones that you will find in other cilia. But tectin 5 is the one that is an expansion that is specific to sperm. So these are all these tectin 5 here. So now I show only tectin 5. And this is the same protein, bear in mind, but you see the same protein actually forms different conformation within the same assembly. So now first I'll show you a normal filament if you want. This is how tectin 5 will form. So these are the red ones. But D and E, for example, they form an interrupted filament. And the reason they are interrupted because they need to fit another protein that is called NME7 in between. Again, it's the same protein. Okay? And then we have cases like pectin G, for example, it repeats only one time in this structure. The others you could see is repeating several times. And F doesn't even form a filament, it's diagonal for a reason where we don't really know. So this is really the same protein. I don't think it's something that AlphaFold can predict. The same protein have so many conformations in the same assembly. Okay, and then I see 3 minutes left, so the question is what next for us? So what I show you is actually only the inside. I show you all these proteins, and then so you should ask me, so what about all the outside? And if you are from the axonal field, you know that maybe some of those are the more interesting one, because these are the ones that have the motor proteins, all these dynames. Okay? So we went about going for this one, but the problem is that I mentioned already that we have the different repeats along the axonal. So this protein that I just showed you, they repeat every 48 nanometer along the axonal. So I put here this box. It's not completely to scale, so don't take me on that. But if I just translate this box, so this is our particle if you want. Yeah? But these proteins that you can see them here, so these are the radial spokes, and they actually repeat every 96 nanometer repeat. So we cannot recover them if we do only the 48 nanometers. So now we got bigger, so it's a bigger box now, which means we needed to at least double our tech set. And all the processing, and this is what we have now. I do not have the time to go into the details of this, but we do have now the atomic model of the 96 nanometer repeat. Again, a lot of new proteins that we identified, and just to convince you that we are going bigger. So these are the numbers. So now I call it a unit cell. There are 181 proteins. Now we are more than 4 million atoms in this model, and more than 500,000 protein residues. And I can tell you that, yeah, the PDB didn't like us last year in our structure, and now they are, yeah, I think like us even less, because, yeah, the position is, not going smoothly, let's say. So I do want to show you, because it is the health session. So what do you get actually from having a structure like that? I think it is amazing and remarkable, because we are not just identifying proteins, we also put them in place. We really say where they are and with which protein they interact, so the whole network. And then what we did, we kind of look at the literature with all these strange name of proteins, and we mapped them on our structure. You see that some of them were already recognized that are part of that were associated with infertility cases, but you can see that there are a lot of grays that were not. So this is, we are providing, if you want, a resource for the field to look for more for diagnostics. And with that, this is my summary. So I think for me it's really a great time to be in the field, because we can do this de novo identification of proteins with cryo-EM, which is amazing. I just show you that even single particle analysis is going more native. And there are more examples, it's not only us. And yes, it is possible to do bigger assemblies, but I think the field can use more tools for model building and refinement. We cannot, for example, refine this whole structure in one go. We have to cut it to 2 pieces, 2 million atoms each time, and even that takes like 3 days or 4 days running on Phoenix. Each part to refine, so it's kind of quite a headache, and I already mentioned the data deposition. And with that, I'd like to finish. I've been really fortunate to have great people working with me, Most of the data I show is from Miguel Liang. I'm very grateful to our local facilities and the collaborators from the veterinary faculties that help us get this sperm from all these strange species. I also want to thank the funding and also thank you for your attention.